things really?
Hey man.
How are you?
Bon appetit.
Smile.
Where do you get this?
Taz gave it to me.
Oh, yo, is there a table or something where we can still look at the presentation? May I land here? No, I can't because you're filming.
He's doing it.
Oh, you're doing it.
Yeah, I'm the cameraman, man.
That's cool. Yeah, I'm just gonna be on the back of it.
Hi, good afternoon, everyone. Good afternoon. I want to invite you to our October version of Books and Bagels. We have some really good graduate students here who are going to present their stories. The purpose for Books and Bagels is to have an interdisciplinary dialogue between different departments. So what we want to do is we want to share our graduate students' research, not just in their own departments or in their thesis when they're writing it, but we want it to be shared by all of the grad students. So please feel free to ask any questions at the end. And I want to invite Jay Butt here, who's the library liaison for the engineering department, and he will be moderating the discussion. So I hope you have fun.
Thank you, Mitch. I'm very happy to see a lot of you are here already today, and this is a great start with the first event here, in this new registered launch. Today, our first speaker is Paul Ullman. He's a PhD candidate in the Department of Biology, and he is a vertebrate paleontologist. And one of those kids that knew he would take his career path when he was 3 years old. He attended Montana State University for undergraduate and took part in many research opportunities while a student there. In his career thus far, he was lucky enough to dig at the first molecularly clean quarry, the quarry where the first burrowing dinosaur was discovered. And he discovered the first and still only dinosaur bones ever found in the state of Nevada. Overall, he has spent 5 field seasons in the mountains of the western US digging onto Tyrannosaurus rex, many Triceratops, and the duck-billed Edmontosaurus. Now, as an NSF-funded PhD candidate at Drexel, he is working on fleshing out fossils involving muscle reconstruction, molecular preservation analysis, and 3D modeling. So here is Paul, and at the end of his presentation, we have one question from the audience, and then we'll follow up with the questions later on. Here is Paul.
First I want to thank Jay and Mitch for organizing this event and for inviting me here today to talk to you guys. And we're going to talk about what I call the art of reviving fossils. In this case, it's kind of synonymous with saying the science of reviving fossils. As Jay was saying, I'm a vertebrate paleontologist. That does not mean that I study humans. That would be archaeology. I do not work on human bones, pottery, anything of that nature. I do, however, work on dinosaurs and other fossil animals. So crocodiles, turtles, fish, those sorts of things. When I attended Montana State, I had a lot of opportunities to do various projects there. This is one of them. This is me at some field work in Montana a few years ago. And here, it's kind of hard to see, but there's a couple of dinosaur bones here on this pedestal rock to protect them, keep them stable as we work around them. And all paleontology starts just like this. We have to start with field work, otherwise we don't have anything to study. But a lot of fun can be had after you bring them back to the lab, too, and that's what I'm going to focus on here. So you dig up your specimen, find something really cool, bring it back to the lab, spend your weeks, sometimes even months, cleaning it up. You can end up with a really nice specimen like this here, a set of vertebrae from a crocodile found in New Jersey, Cretaceous age. When I talk about the phrase reviving a fossil, what I mean by that is what are the steps behind the process and what's the science involved in going from a set of just bones to something like this, a fully fleshed out model with skin, eyes, things that don't preserve, but we can still stagnant, static model, what's the science evolved heading towards? Something like this. So, I only have 10 minutes, but I think this 1 minute is really important. This minute captures what one of our end goals is now in paleontology. It's to recreate an extinct organism in motion, in its environmental setting at the time that we can infer from the rock record, interacting with other species we find in the fauna preserved at the site, and in a manner that all of you can relate to. You don't have to be a paleontologist, you don't even have to be a scientist. You can remember, hey, on Discovery Channel, I saw a crocodile acting like this. So, this is an animation made by one of my coworkers in our paleo group a couple of years ago. And we are now trying to do this with another species, a new dinosaur that we're describing in the Drexel paleo group. It's a sauropod. It's like Littlefoot from Land Before Time, something like that. Long neck, long tail. Looks something like this. If we reconstructed it artistically, something like the drawing at the bottom here. Now what we're doing with this skeleton is we are using a 3D laser scanner to capture a 3D model of every single bone. And with that, we have two goals. First, to articulate the bones back together in the digital realm as a skeleton, and then a fleshed out model, and eventually an animation like I showed you a second ago. And second is to use a 3D printer that will print out each model to scale that we can use to build a working robotic skeleton of this dinosaur with a lot of help from the engineers, of course. So I'm going to walk you through the process here of what we're starting. Step one, of course, find a specimen, dig it up. So this is my advisor, Dr. Lacavera, with Ullman, one of the forelimb bones here of our new dinosaur. And this was found in 2004 in Patagonia, Argentina. We're still working on deciding on a name. We'll have that soon. So you collect it, wrap it up in a plaster jacket, like you would if you broke your leg, something like that, exact same way. Bring it back to the lab, and after some weeks to months of painstaking cleaning work with brushes, dental picks, X-Acto knives, a lot of putty to infill the cracks, you'll get a very nice specimen like this one. Our next step in the process then, bring in the laser scanner. So it doesn't look like much, it's just a little gray shoebox-sized thing down here, but it traverses 4 red laser lines there across the surface of the bone, and it will pick up an individual scan that you can sync together many of those to make a full model of each bone. And this is an example of one right here. This is the Humerus Upper Arm bone that I just showed you a second ago. So, looking at this right away, you can see some really useful data. First, you can tell details about breakage and deformation up here to the bone. And maybe one of the most striking things is just how well you can tell the depth and the shape of this bone from this appearance. So even though it's only one bone at this point, it's still very useful to us because it offers a way to standardize our scientific illustrations. Normally, we would call in an artist to look at the bone, draw it for us, and there'd be some subjectivity as far as where the artist wants to shade, how dark or intense he wants to shade in various areas. With this, we can capture a still of the bone in certain anatomical layouts, and we have a fixed artificial light source shining on the bone. And we can do that the same for every single one and make it completely standardized. So no more subjectivity. Of course, the next step that we can do is bring these bones together. And we haven't yet mastered how to capture an animation of like the elbow in this case moving. We're working on that. It's a slow process. We normally work with bones, not with application, you can see some things about how we can infer the articulation at the elbow joint here. Should the bones be twisted or rotated a little bit more? We can test this out very easily in a digital program, Maya in this case, as opposed to moving around these individual bones which each weigh over 300 pounds. In the end, we want to use those to make our digital skeleton articulated and walking in motion. And the other venture was this 3D work, was the 3D printing I mentioned. And with this as an example, you have a full-size humerus from a dinosaur of a similar type on the left, and on the right, a 1/10 scale plastic printout. And I brought one of these here today to show you guys what I'm talking about. So this right here, one of those printouts, is the fibula, or a lower leg bone, from our new dinosaur. It's not a clay artist sculpture, anything like that. It's an exact copy, 1/10 scale. It's perfect enough, really, that I can take this home at night, work from my bedroom if I wanted, describe the anatomy of this bone, without ever having to look at the full size bone, which is about this tall. That's there. So,
How much does that depend on the one individual you got? Could you tell that this particular animal had some sort of health condition, or is it just a standard for that species?
well, first thing you have to do is judge how old your particular dinosaur is. We have one individual for our new species, and you can look at the microstructure of the bone in cross-section, and from that data you can tell if it was a full adult, if it was still growing rapidly. Our data says that it was almost a full adult, so very close to that. The second thing is when you look at the bone itself, you can tell from fracturing or breakage or offset of different things how much the bone is deformed. And for many of our bones, we're very lucky. There's very little deformation. There's no big cracks, offsets, smashed edges, anything like that. So for our working digital articulated skeleton, if we want to make it walk, we obviously have to work with the muscles. And for that, the set of muscles that would be present for dinosaurs, for example, are essentially the same set that are in us. They're in common with almost all vertebrates. So this being some other groups' work in Tyrannosaurus rex, but it's something that we want to work towards in the near future here, doing almost the exact same thing. Figure out where the muscles attach based on scarring they leave on the bones, what relative sizes they should be, and at what angles they act, and use that to work towards making a fully robotic model. And we're doing this project in cooperation with the biological engineering group over here in Randall Hall, Dr. Tangora's lab. This is one robotic model that they've already built, a robotic fish. What they were doing with this is they built these fins that can undulate in different patterns and speeds, and they wanted to test the energy efficiency of swimming at different locomotion styles. So for our robot with them, we want to test various hypotheses on the energy efficiency of locomotion styles at different speeds, at different stances, how wide should the limbs be apart, at what sort of angle should the leg be turned, those sorts of questions. Now, to shift gears, there's a second way that we can get to reconstructing fossils, and that is by preserved soft tissues in the fossil record. And this is in delving into a realm that we're now terming molecular paleontology, just begun in the last few years. For my work specifically in this realm, I'm using a case study site, CHS bone bed in South Dakota, and it's a bone bed with thousands of bones of this duck-billed Mosasaurus here, around 70 million years in age. So a great sample size to work with here. And your first step in these sorts of analyses is to take a little bit of the sediment that the bones are preserved in and expose it to a weak acid that would eat away calcium. What you do, or after you've done that, a week or two in this weak acid, you'll see that things like quartz, sandstone here, are left behind and just kind of looks like little speckly grains. These larger black blocks here are probably bits of coal or organics that were in the sediment. But there's no branching, complex, real biological structures here otherwise. Now when you do the same thing to a fragment of dinosaur bone, totally different. You can see it's parallel, anastomosing network of light-colored tubular structures. And if you had done this with a modern piece of bone, this looks exactly like the blood vessel network running through bone. And in addition to these vessel-like structures, there are these little tan fibrous areas that look a lot like the proteinaceous matrix of bone. And it has, in many cases, these little cell-like bodies that are often parallel aligned. And once again, if this was modern bone, you would call those osteocytes, bone cells. They still have, in this case, some of these little Y-shaped processes that would connect to each other. What we can do with these various tissues and cells is run molecular biology assays on them, characterize their proteinaceous character, get protein sequences from them even, and that sort of data can help us narrow down things about, for instance, appearance, where and how large would keratinous sheaths be on their claws or on their beak, something like that. We could also look at ideas about physiology, such as metabolic rate, how fast or slow that would be. When we combine that with the 3D modeling of robotics, with new information on potential behavior and locomotion styles, maybe even means of reproduction, we can really add a lot of scientific background to these sorts of reconstructions. Just like even this drawing here. Is this stance of this leg feasible? We can test that now. So with that, I'd just like to thank you guys again. And please come by and visit anytime. We're always happy to have visitors. We're over in the new PISP building on the top floor, 504. And we'd love to give you a tour. Thank you.
Well, we have time for one question right now, and then we'll have in the end. Yeah.
I have a question about the last section where you did the molecular analysis. So whatever cells you are seeing on in those micrographs, how do you make sure that those cells actually belong to the dinosaur? Over thousands of years, when these bones were lying in ground, maybe there were some parasites growing in there, and you see those small little cells. Actually, maybe they are not dinosaur at all.
Right, that's a very good question. So, if our hypothesis is that they are original to the bone itself, we would expect them to have the same appearance as the same structures in bone and the same chemical nature. These sorts of cells and tissues are very conserved. They're kind of in common to all bone. If you have bone, you have those sort of structures. And so, if we run an analysis with antibodies that attach and respond to only certain types of proteins, we can identify, is there a portion of this protein in this cell? And in many cases, when that's been tested, we have found the exact same binding pattern as you find when you run it on modern bone. When you run the test with antibodies that would bind to irrelevant proteins that shouldn't be there, or ones that are made by bacteria, we haven't yet seen binding. So we have pretty strong evidence that says that they're original. It's very, very good.
Thank you, Paul.
Thank you.
Our next speaker is Luminous Kinnaman. She is a PhD candidate in civil architecture and environmental engineering here. She got her master's in civil engineering, with specialization in coastal engineering at Leibniz University in Germany. Afterwards, she worked for 2.5 years at the Federal Waterways Engineering Research Institute in Hamburg, Germany, where she was in charge of improving the Port of Hamburg maintenance dredging plan by using numerical models. Most of the research was always related to simulating sediment transport in rivers and coastal areas using different hydrodynamic numerical models. She worked at first as a teaching assistant for fluid dynamics, hydraulics classes at Drexel, and is now a research assistant for the Philadelphia Water Department. They do hydrodynamic numerical modeling of the Delaware River around Philadelphia as part of Green City Clean Waters project. So here is Lamana. Please welcome her.
Hey, this is hard competition. How can I compete with dinosaurs? So I do my best. So already said I will talk a little bit about modeling of marsh systems in the Delaware Bay. And here in the background you see a little bit of our aerial picture where you can see from the bay red tidal channels are meandering through the through these tidal marshes, which are pretty much wetlands that inundate as they get regularly flooded and dry during the tidal cycle. So what I will do, I will tell you a little bit about the Delaware Bay marshes, why we do this research, why it is important for everybody, not only for the coastal engineers. So it's a very important ecosystem that everybody should be interested in and take a look at if possible. And we'll also show why interdisciplinary research, which is one of the topics that what we're talking about here today, is a very good example in this case, because I have to go into very different fields to get information to make my model work. And again, I will show you a few of my model applications that I've done so far, but there's still a lot to do. So just a few glimpses. So where are we, first of all, when I say talking about marshes? We're here in Philadelphia, and you see that we are all the way up in the Delaware Bay estuary, which from this distance, seen from this distance, doesn't look very dramatically interesting, relatively regularly shaped compared to if you know the Chesapeake Bay down there, which all these tiny little coastal features. So from far away, the Delaware Bay doesn't look particularly interesting. But if we zoom in, we see a very special feature that even worldwide is very rare. If you look at the coastal lines of the Delaware Bay, you see that almost the whole bay is fringed by a connected network of tidal marshes. You see, and if you would zoom further in, it's almost like back to blood vessels and network, it's almost the same. You see that the tidal channels are eating into the land and go finer and finer and finer. So these are very intricate systems, and this is what makes it so difficult and why there aren't much modeling efforts have been done by now. So why are they important for us? As I said, first of all, we should try to preserve any kind of feature that is relatively rare, even worldwide. So, but also, if you look at wetlands, they provide a very important service to our bigger water bodies, the rivers, the bay, and they provide a real good resource and habitat for all kinds of flora and fauna. So, in terms of health and function, they kind of work like kidneys to a body, because water with flood tides gets into the marshes itself, and there all these harmful, potentially harmful materials get filtered and preserved there. And what not many people know, they also store a lot of carbon. So everybody always talks about carbon storage in terms of forests and trees, which are important, of course, but worldwide wetlands store about as much carbon as forests do. And the bigger problem is, because this carbon is stored underwater, as soon as these wetlands degenerate and fall apart, this carbon is released back into the atmosphere. We all know that that isn't good. As I already said, it's a home of a variety of different animals, many of them threatened. And we have all kinds of mussels, crabs, fish, birds, and I'm happy to say that in the recent, say, 10 years, we see many of these, especially freshwater mussels and oysters, coming back. So all these environmental efforts that are being done seem to help already. And also, it is one of the biggest and most important resting places for migratory birds. So when the birds in winter go from Canada and wherever in the north down to warmer places, they stop over here at the Delaware Bay because we have an abundant resource of horseshoe crabs, and somehow the birds like to eat their eggs. So, but same thing, if we use our wetlands, they lose their food resource. But it's not only good for plants and animals, it's also important for us. You can imagine, very good timing, on Monday we seem to be hit directly by a Category 1 hurricane, possibly. The models look like it is going to happen. And these kinds of strong storms, but also smaller ones already, like typical winter nor'easters, they give you a lot of wave activity. But this wave energy doesn't really reach our dwellings yet because there is this broad band of marshlands where all that energy is taken away. So they also buffer us against coastal storms. And in the end, of course, this is a great recreational area for everybody. So for hiking, for canoeing, fishing, hunting, you tell. The sad part is that you've seen the picture, the urban corridor from Philadelphia down to where the bay opens, there's only barely 5% of the pre-settlement wetland acreage left. The problem is that as soon as we start building, back in the day we didn't know they were doing anything good. They were also always considered like useless swamps, let's fill them in and use them for something else. And the other problem is also that rising sea levels prevent them from migrating. You can imagine if you have just open space, sea level rises, the deep water part falls apart, but what is newly flooded can turn into new marshland, no problem. But nowadays, with all these structures that we build, the marsh can't go anywhere. So that's why we lose so much, and this is why we are happy nowadays to know about it and try to find different solutions for our structures to build, for example, or if we build at all. So why is this so important interdisciplinary? As Jay already said, I did a lot of sediment transport in deep water areas like rivers and coastal areas. And there we have hardly any interaction with obstacles. There is a little bit of underwater plants, but they are not that important for flow because we've seen earlier in the little movie that these seaweeds, they go with the flow. So, but in wetlands, we have this flow through very stiff and dense vegetation, so this is not going anywhere with the flow. It doesn't care, but the flow, the water has to go around that one. So this is a big difference in traditional hydrodynamic modeling. In rivers, we have a well-defined sandy bottom, and we have good physical equations that describe how sediment transport happens, when it starts. Not so in wetlands. The problem is mussels and crabs have an influence on the sediment. So, for example, some kind of mussels form dense packs and produce some glittery substance that kind of glues the sediment together so it doesn't fall apart on the roadside easily. On the other hand, I don't know if you have been out there, if you've seen these little fiddler crabs with the one big, clam. They dig through all these wetland grounds and make it a lot more easy for that sediment to be eroded away. So the problem is now I can't, as an American modeler for water, for flow, I can't ignore these effects anymore. But actually I don't know much about vegetation, about the life cycles of mussels and crabs and what the hell they are doing. So that's why we need to approach people from other fields, especially in my case now, biologists, ecologists, but if you go a further step and are interested also in water quality, there's also a lot of chemistry that comes into part. So many possibilities now that different kinds of research overlap quite a bit. So just a little example, you see there's a lot of vegetation, I just can't ignore it. So this is a typical marsh down in the Delaware Bay area. So why do we do this numerical modeling? The problem with marshes is they are really large and very hard to access, because I tried once to get out of the boat and take some measurements. It took me 3 steps until I landed in the mud, because if you don't move fast enough, you get stuck and sit down. So field experiments are difficult, costly, and very time-consuming. And also, you can imagine you can't be at the same point at different points at the same time. So you will always get only snapshots of one specific period in a certain location at a certain time, but no synoptic data. So in this case, numerical models are practical because if we can produce close enough conditions to reality, we can take a look at any detail that we want at the same time. Also, I told you about all these stupid things that humans did. We thought back then it's a good idea to, as I said, just dry out wetlands and stuff, but we didn't know about the long-term effects. So nowadays, before we make any changes to the system, we could do it first in a model and see what happens. And if it's good, great, then we go and do it in reality. If not, then we know we prevented some big trouble. So in the end, if this at some point works in a very efficient way, this is what we're still working on, it's a very good tool to better understand and to help maintain and restore wetlands. So one of my model areas is around the Drexel Creek Reserve. You see this is part of the Delaware River, and I just roughly outlined the water of that marsh. You see that it goes down from relatively coarse model elements of 300 meters down to 3 meters. This is the biggest problem we have. If we want to resolve this area, which has even only the second order tidal channels are still about only 10 meters wide, and you can imagine at least 3 elements to at least have a trapezoidal shape so that you can put some water in there. We have to go down to very small elements. And that's nowadays, it's getting better because our computers are getting better, but with a model with 260,000 elements, I am barely faster than reality, so it's not a forecasting model, even with the supercomputer from University of New York. So, but still, it helps us to see how these processes in there work. So, the picture you've seen before is from this area, and the reason I picked this one was because it's one of the recent reserves in Delaware, which provided me already with at least some data. That's the other big issue. You have a nice model, but how do you feed it? So it's very difficult to get initial conditions. My boundary conditions I just produced with a bigger model of the whole estuary, just as an example. 260,000 elements for this tiny area. My whole Delaware estuary model has roughly 19,000 elements. So there's no details. That's why I can spend a lot, can go a lot bigger and be a lot faster. As one of the examples I want to show is sediment transport. We are interested in erosion and deposition, and with a very simple test, we showed very clearly that you can't ignore vegetation. In this case, I used all the areas in between the channels are the tidal flats, and at the moment I just assume a higher roughness on areas with vegetation with low roughness where you just have sand. And this is all the changes we did, and we see, of course, expected at the corners you have some erosions where it can attack the sediment, but you can see that where we have the high roughness because of the vegetation, there's hardly any changes. And this is only after after 3 days. And this is, as I said, what you would expect. On the other side, if we just assume there is no vegetation, after a couple of days there wouldn't be much left of the tidal flats. So that was a very easy, quick test. My other model area, from where you look, is National Wildlife Refuge. And we started this one because they have some real trouble there with losing wetlands and they can't explain to themselves why. So it was ponded and open water areas inside get bigger and bigger over the years, so, but they don't know how. So they asked me if I'm making a model already, so I would make one of this and we could make some trial and error tests. This is especially exactly the kind of example I had from before, where we can change the marsh geometry without doing actually anything in the real world. One of the theories is, you see here, all the brown ones is a dry, we are now in a F cycle. So this is the dry area, and here are the open water ponding areas that get bigger. One of the theories is that this apparently man-made ditch that connects the inner channel to the bay might transport sediment out of the marsh instead of keeping it inside. So what I did, I just made one example with the channel, and the next step without, and look what happens to the stream. So here we can see now apparently, because of the density, the vectors get bigger with high velocities. It's quite an active channel, an important channel for flow. And when we remove it, this whole area calmed down a lot more. So it definitely has some influence on it. On the other side, we looked before, now this channel gets more important. If I flip back and forth, you see that. So it could, of course, mean that net, the sediment transport out of it, is the same. But we definitely have shown that changing this little detail changes the flow situation totally. So this is just at the beginning. These results came just in yesterday. So a lot more to do. And the same thing also for our flood situation. And we see the same here. So more flow through the channel and in that more activity inside that area and less when we remove that channel. So overall we see it's hydrodynamic modeling, yes, but if you go into marshes and any other kind of vegetated areas, we have to take it into account. And there we go back to the interdisciplinary research where we know now we have to go to other people to find out how these behave seasonally or what kind of condition they need to survive or die. Some of our changes might change the water level so that the plants will die off. So, and what I didn't talk about is it's even for social sciences, it's very important nowadays. There are many, many things going on that educate the general public about the importance of wetlands. And it only works if we can preserve them when everybody knows to appreciate them. So there's still a lot of room to find out how to better how to do some public relations for wetlands so that people like them. In the end, thanks for all who gave me money and let me use this for computers. Thanks a lot. Questions?
Thank you, Ramana. We have a time for one more question and then we'll end up later on with more questions. Anyone?
What sort of time scales are you modeling?
As I said,
Hours?
I go for days, but because I'm roughly, especially when I turn on the sediment transport, I'm happy if I am in a timeline of 1-to-1, that's the model time, this is fast in reality. So we are looking for modeling, I got a 2-week to month timeline. So this at least gives us the ideas to know about the spring tides, so that we have one neat spring cycle covered, all possible flow situations that could arise. So it's covered with 2 weeks. So.
Thank you, Ramana. So our next speaker is Brooke Mary. She is a PhD candidate in the marketing department here. Her passion for marketing started when she was 16 years old and read an article in the Philadelphia Inquirer which explained how the more symmetrical one's face is, the more attractive others perceive him or her. From there, she began questioning how underlying psychological mechanisms influence marketing and was a double major in marketing and psychology at La Salle University. While at La Salle, she worked as an intern in the Marketing Research Department at Commerce Bank, now TD Bank, which turned into a federal position, and she accepted a full-time position as a marketing research analyst. While at Commerce, she went to Temple part-time and received her master's in marketing, consultation marketing research. After Temple, she switched from the client side to the vendor side and worked for TNS, the largest custom marketing research firm in the world, as a senior project director. After TNS, she worked for Hard Hanks, the largest direct marketing firm in the US, as a marketing research manager. At that point, she decided to take a break before going back for her PhD, and it took a year off.